How many different CFTR mutations are there? Imagine a gene that holds the key to understanding cystic fibrosis, a complex condition affecting many parts of the body. This gene is called the CFTR gene, and it can have many different changes or mutations. Currently, scientists have identified over 2,000 distinct mutations in the CFTR gene that can lead to cystic fibrosis. These mutations come in various forms, such as changes that stop the gene from making any protein, mutations that cause the protein to fold incorrectly, or ones that affect how the protein functions as a channel in cell membranes. Since the discovery of the CFTR gene, research has uncovered more than 2,100 mutations worldwide. Some mutations are very common, like the delta 508 mutation, which is found in about 66% of cystic fibrosis cases globally. This mutation causes the protein to be misfolded and degraded before it reaches the cell surface. Other mutations are much rarer and tend to be specific to certain populations or regions. For example, some mutations are more frequent in European populations, while others are seen mainly in certain indigenous groups or ethnicities. Scientists classify these mutations into different groups based on how they affect the CFTR protein. For instance, some mutations prevent the protein from being made at all, called class I mutations. Others, like the delta 508 mutation, result in a protein that is made but doesn't fold properly, which is class Roman II. There are mutations that cause the channel to not open correctly, called class triple, or ones that make the channel conduct ions poorly, known as class Roman IV. Some mutations reduce the amount of protein produced, called class V, while others make the protein less stable on the cell surface, called class Vi. Understanding these classes helps doctors and researchers develop targeted treatments that address specific defects. Because of this vast diversity of mutations, genetic testing for cystic fibrosis often focuses on the most common mutations relevant to a person's ethnic background. Testing panels are designed to include these mutations to improve detection rates and keep costs manageable. Knowing the exact mutation in a patient is crucial because some newer drugs, like ivacaftor or lumacaftor, are designed to work only on specific types of mutations. In summary, cystic fibrosis is caused by a wide array of mutations in the CFTR gene, with over 2,000 variants identified so far. This genetic diversity impacts how the disease is diagnosed, how genetic counseling is approached, and how treatments are tailored to each individual. If you or a loved one are concerned about cystic fibrosis, it is always best to consult healthcare professionals who can provide personalized advice based on the latest scientific understanding.